This is Chapter 13, Gene Expression and Regulation. So the chapter starts off with looking at a genetic disease. So we are going to be talking about the expression of genes or DNA and how that relates to phenotypes or traits of an organism or traits of a cell. And cystic fibrosis is a fairly common genetic disease as genetic diseases go. Most of them are pretty rare, but this is a more common one. We'll get a little bit more into detail about it as we move through the chapter, but to begin with, number one, it is a recessive allele, and this will make a little more sense as we get into later chapters, but you might get some understanding of it now. Recessive just means that you need two copies of it. So we each get two copies of every gene or allele. We get one from our father and one from our mother. And so to have cystic fibrosis, you have to get this allele or version of a gene from both your mom and your dad. So most of the time, people will just be a carrier. They'll have one bad copy and the rest of, and they basically don't have the disease. But if they do have the disease, it means they have two copies. They got a bad copy from mom, got a bad copy from dad, and what the gene codes for is something called the CFTR protein. And CF stands for cystic fibrosis, TR for transport protein. And it's a chloride ion channel, as shown to the right over here. And the one on the left, so this one is the one that is functioning correctly. You got two bad copies, as I said, homozygotes. Again, we'll go over that in a later chapter in detail. Homozygotes experience a buildup of mucus in the lung, which allows bacteria to infect the lungs, and eventually they generally do not make it. So the life expectancy is much reduced. So what is the normal functioning of this ion channel? Well, basically in the lungs, so hey, I got a handy picture of the lungs down here. That's a healthy lung, and that's a healthy lung here. And the lungs tend to sort of accumulate stuff. One, as a necessity of simply breathing, dust, small particles get into the lungs, and we have to remove it. And the way that we remove it is we make sure that the coating of our lungs and other tubes in the lungs basically are covered with a layer of water and mucus. So one of the mechanisms by which we accomplish that is we take these ions. So the blue things right here, I'll circle a couple. Those are representing chloride ions, and we pump them out. So we pump them onto the surface of the lungs. What happens after that, which the book doesn't really talk too much about, is when we get a higher solute concentration outside of the cells that line the lungs, what happens is water follows. So H2O is going to diffuse out of the cells and onto the surface lining the lungs. What that does is it makes the mucus that lines the lungs a little bit less thick, a little less viscous, and it is more easy for the body to remove. Now, if we don't have a functioning chloride ion channel, as shown on the right here, what happens is chloride ions do not diffuse out of the cell and on into the area that is kind of lining the lungs. And so what happens is water doesn't diffuse out either. And so the mucus is just much thicker, and it can't be removed as easily. And so bacteria and well, mostly just bacteria will get into that mucus, and they can start growing and cause various infections. The mucus itself, when it gets to a certain degree of thickness, it can obstruct the airways as well. Shown down here in the bottom right, cystic fibrosis. You can see this could be a bronchiole or some other tube that is carrying or transporting air through the lungs. It just gets more narrow because there's more mucus and all sorts of other things. The central dogma of genetics. So this is what we're going to be talking about this whole chapter. DNA, that is where we hold genes. So the genetic information is held in DNA, stored in DNA. And DNA is in the nucleus. So this down here is a very simplified version of the cell. So in the nucleus, that is where we have DNA, and that is where transcription occurs. That's going to be the first major topic we'll talk about. Transcription converts DNA to messenger RNA, or mRNA. M stands for message. And then the messenger RNA leaves the nucleus and goes into the cytoplasm of the cell. Yay, we've got that word circled down here, cytoplasm. And in the cytoplasm, by ribosomes, the messenger RNA is essentially read, the message is read, and some type of polypeptide is produced. And a polypeptide is a bunch of amino acids strung together in whatever order the DNA instructs it to be assembled in. Little bit of information about DNA versus RNA. So on the left is deoxyribonucleic acid. We talked about that in a previous chapter, and now we're going to compare it to ribonucleic acid. And there are a couple of differences, and we do need to know all of them and remember all of them. The first one is the most minor and insignificant. So the sugar in DNA is deoxyribose. So that means it's missing an oxygen, and it's missing an oxygen right at this second carbon right here. That's why it's called 2-deoxyribose. It's the second carbon. We like to number our carbons. 1, 2, 3, 4, 5. And now let's look at the right-hand side of the screen. So this is ribose, so it's not 2-deoxy, it's just ribose. So ribose has an oxygen on that second carbon. So that just makes the sugar a little bit different. And this is one of the structural differences that is actually not going to play much of a functional role, but every course I've ever taught requires that you know this, which is sort of, I don't know, whatever. The remaining details of the differences between the two are structurally important, functionally important. So in DNA, there is a double helix shown right here. That's a double helix. And in RNA, it is simply a single helix, a single helical structure. Single-stranded is the most important part there. RNA is super similar in terms of the type of information it carries. So DNA has C's, G's, A's, and T's. Oops, I kind of cover the T there, and T's, and RNA has almost the same bases. It has C's, G's, A's, and a U instead of a T. The RNA can also do base pairing. So in DNA, we're going to have A to T and C to G, and in RNA, when it does base pair, it is going to have A to U because it doesn't have a T, and then C to G just as in DNA. Note that U is also a single ring structure so that the base pairing kind of still matches up a double ring and a single ring. Transcription and translation, another just sort of big overview. So again, in the nucleus is where we have DNA. And now that we know that RNA is a single strand, what happens is in the nucleus, transcription occurs. And we transcribe a gene, and it produces an mRNA with a nucleotide sequence that is complementary to one of the DNA strands. So in the last slide, we mentioned that that there is some base pairing rules. So if the DNA strand here was A, C, C, G, for example, then the mRNA strand would be complementary to that. And we're going to go over this in greater detail later, just an overview now. So if DNA was ACCG, then the mRNA would be U, because A pairs to T in DNA, but U in RNA. So A to U, C to G, C to G, and G to C. All right. So this sequence would be in the mRNA. mRNA leaves the nucleus and goes outside into the cytoplasm where ribosomes, shown right here, are going to read the message in the mRNA and produce a protein. A couple of examples of types of protein products, or as we've written over here, polypeptide products. Polypeptides are proteins. There are some minor differences, but they're basically the same thing. So remember that DNA, the only thing it does really is code for proteins. A couple of examples that we've talked about already. Let's see here. This one right here would be a transport protein. That's a channel protein involved in facilitated diffusion. This is another protein. This would be, looks like ATPs involved, so active transport. Other proteins will be involved in connecting one cell to another cell, and we could just keep going on and on and on with the functions of proteins, because that's what we are. DNA codes for proteins, and that's what essentially distinguishes us from trees at a molecular level anyway. True though. Another example, DNA codes for polypeptides of DNA, RNA, polypeptide. the example here is skin color or skin pigmentation, excuse me. And there's 3 types of polypeptide that can be produced here, and they're all types of melanin. And you don't need to memorize them. They're heteromelanin, pheomelanin, and eumelanin. And they contribute, they don't determine all of the person's skin color, but they contribute to skin color. Could be darker, could be lighter. And then there are even more genes that are going to contribute to this. And even the environment will contribute to this. If you've ever gone outside in the sun for a while, you'll notice that the next day your skin might be a little bit darker. You will get a little more tan, and that's because cells also have the ability to do what's called upregulation of a gene, so we can use the gene more often. So if the cell gets a signal from the environment, such as we need to use this gene more often, to some extent, we can just use the gene more often. Types of RNA. So DNA is transcribed into mRNA, and the top one here, that is mostly what we're going to be talking about, because this is the sequence of mRNA nucleotides that carries the genetic information out of the cell. And the base sequence here is going to determine the type of protein that is produced. However, the remaining molecules of RNA have a couple of different names. One is called a tRNA here, and then in the ribosome, there's something called rRNA. There it is right there, ribosomal RNA. And we can always think about the function of RNA being related to the fact that it can complementarily base pair with other nucleotide sequences. So the tRNA here is going to be involved in something about the genetic code, matching up one bit of mRNA with a amino acid. And the ribosomal RNA is something we're not going to talk much more about. Suffice to say that it is part of the ribosome and helps the ribosome recognize messenger RNA. So this ribosome is eventually basically going to bind to mRNA. So there'll be a sequence of mRNA that goes through it. I'll draw a sequence now. Okay, so there's the mRNA going through it. And the ribosome needs to be able to identify where to start on that message. So where's the beginning of the information in mRNA? Where does it start? And tRNAs and tRNAs and ribosomal RNAs, they play a role in that, and it'll make much more sense when we get to this topic, which is translation. Right over here, this is a zoomed in, more detailed picture of a tRNA. You can see they're sort of shaped like a T, but the T also stands for like transfer, because right here there would be an amino acid of some sort, and the tRNA is going to be involved in transferring that amino acid to a growing protein. Right down here is the more important part, and that is what's called the anticodon, and AAG means that it's going to complementary base pair to, let's see, AAG, that would be UUC, and that'll be on mRNA. More on that later, just kind of an introduction for now. So the genetic code. Now looking in detail at what information is stored in DNA. So DNA, the informational unit in DNA is in sequences of 3 or groups of 3 nucleotides. So I'll just kind of separate a couple here. This is one group of 3. It looks like that's GCA, and then the next group of 3 looks like it is TCT maybe, no, TGT, and so forth. So groups of 3. What is the meaning of these groups of 3? Well, essentially, each group of 3 DNA bases is going to code for a single amino acid. So that's down here. This ARG stands for arginine. And how is that coded for? It's coded for in messenger RNA, and that's where the real kind of code of the genetic code is. So let's take a look at DNA to mRNA. So the transcription process, where the nucleus or something in the nucleus reads the information in DNA, basically results in, if this up here is GCA, so it's a little hard to see there, GCA, then a complementary sequence of messenger RNA is going to be produced. So CGU. So G is complementary to C, C is complementary to G, and A is complementary to U. Same thing continues with DNA over here. The next sequence of 3, so TGT, so T is complementary to A, G is to C, and T to A again. So we get this next group of 3 messenger RNA molecules or bases. So in mRNA, a group of 3 is called a codon. And codon essentially stands for a code. There's a code in that group of 3. And to the right, this is the universal genetic code. So let's take a look at CGU if we can find that. So first we'll take a look at C, that's the first base, and then C, what was it, CG. G is in the upper right, so CG and then CGU. CGU right here. So if you can see where I'm circling here, CGU codes for arginine. That's an amino acid, and it'll be part of the polypeptide. Next one, coming back to our sequence of mRNA, ACA. ACA codes for threonine, and again, you can just go find it on the genetic code, ACA. So A, C, A, and I found it. ACA codes for threonine, and that's how you figure out what the sequence of amino acids is going to be in a polypeptide. The reason that we say it is a universal genetic code, it's universal because all living things use it. Whether we're talking about a banana, a bacteria, a plant, an animal, whatever, everything uses this exact same genetic code. And this is one of the strongest pieces of evidence that all living things have a common ancestor. We all use the same genetic code, exactly the same. And if you'd like to take a crack at decoding the message in messenger RNA, well, I already did this one for you. So what about the sequence at the bottom? So what would be the amino acid sequence from the following mRNA sequence right here? So starting is AUG, then ACA, then AAA, then GCU, and so forth. You can pause the video and see if you can figure it out. Pause it now because I'm now going to show you the answer. All right, so if you didn't pause it, you lost your shot. Here we go. Here's the answer. AUG codes for MET or methionine. I'll just do one of them. A, U, and then we look at the intersection between those two. AUG, so there it is, MET, methionine. And if you're wondering, do I have to memorize all those different amino acids? Nope, don't have to memorize them. We will now look at transcription in greater detail. That's where we're going to go from DNA, use it as a template to make mRNA. Carry the message or the information in DNA out of the nucleus. It occurs in 3 phases: initiation, elongation, and termination. And I'm not going to be asking you any questions about trying to mess you up in terms of what's the difference between initiation and elongation. I'm really going to be looking for, do you understand the entire process of how information is converted from DNA to RNA? So initiation is when we have this enzyme called RNA polymerase. And RNA polymerase basically does everything. It's going to bind to a promoter region, and we'll come back and talk a little bit more about the promoter region a little later. The promoter region, let's say, that is right about here, sort of. It's like more or less there. RNA polymerase binds there, and we have a special name for where transcription starts, because you might wonder, DNA is extremely long. There's millions and millions of base pairs, so where do we start? And there are a whole bunch of other factors. They could even be environmental factors, such as when do leaves fall off of a tree in the autumn, in the fall every year? Well, that happens to be generally in most species day length, sometimes temperature as well. And all these things can cause various changes in regions of DNA called promoters. So right there, change in daylight could cause something like another protein to bind there, which will facilitate the binding of RNA polymerase. So a little more detail than you actually need to know, but that's what a promoter is. It promotes the initiation of transcription. It promotes binding of RNA polymerase to the start of, in this case, gene 3, as opposed to gene 2 and gene 1. Gene 1 and gene 2 would also both have their promoters. Each and every gene pretty much has its own promoter. There are exceptions though. So what happens after RNA polymerase has bound is that it separates or unwinds the double helix near the promoter, as you can see here. It has been unwound from top and bottom. It's been unwound. Elongation, what happens now is RNA polymerase, that's this enzyme right here, RNA polymerase, is going to move along the DNA strands, continuing to separate it, and it's going to synthesize a strand of mRNA, or mRNA there, that is complementary to this strand called the template. So the template strand is going to be complementary to the mRNA. For example, if the template strand down here is CGT, then the strand of mRNA will be GCA, complementary to the template strand. Here's I think, a more detailed view of elongation. This is the same thing, but now we can actually see that the template strand, let me point that out to you. Here's the template strand. It's the one on the bottom. So this is the template strand. And it is going to be complementary to the messenger RNA that is getting synthesized. The other strand up here is going to be identical to the mRNA that is being synthesized, and that's why it's called the coding strand, because the code that is in that strand of DNA will be the code that's carried out of the nucleus. So why is that? Well, essentially, take a look right here. So C, so same code, right? And A, same code. U, T, remember RNA doesn't have T, so U will replace T, and then C, C. So the coding strand is 100% identical in terms of the sequence of bases compared to the RNA, compared to the messenger RNA in this case. So that's why we call it the coding strand. Its code is carried out of the nucleus. Termination. So termination, basically, we're done. We reached the end of the sequence in the DNA, and messenger RNA, this strand that has been synthesized here, is going to disengage from the DNA. RNA polymerase right here, RNA polymerase is going to disengage from the DNA. The DNA will wind back up into a double helix, and that's essentially it. How do we know when it's at the end of a gene? There's a termination signal, and it's just a sequence of bases, basically. So good enough. End of the gene, the messenger RNA carries all the bases that are part of that gene. Conclusion. I already basically said this. So the RNA polymerase right here disengages from the DNA, and the RNA itself, and we're going to be mostly talking about messenger RNA, so I'll put a little N there. The messenger RNA also, it's been produced, and it's just going to be transported out of the nucleus for the next stage of the expression of this gene. And again, here's just a more detailed diagram of what's going on in transcription. You could pause the video and try to describe what is going on here for practice. Also, if you have not watched the link to an animation for transcription, I would do that now, just because it's helpful to see the whole process moving and getting done in kind of animation form. So the messenger RNA is going to act as the intermediary by serving as the complementary copy of a DNA gene that is read by a ribosome. The ribosome is the cellular machine which reads the sequence of codons on the messenger RNA and then places the appropriate amino acids in the correct order and binds them together to build a protein. Ribosomes can occur free, floating, or dissolved, I guess, in the cytosol. Could also occur on the rough endoplasmic reticulum. In eukaryotic cells, each gene consists of multiple segments of DNA that encode for protein, called exons. Exons are interrupted by other segments that are not translated and don't have anything to do with the final protein. These are called introns. So the messenger RNA must be processed before it can be translated at the ribosome. That's what we'll look at now. Take a look at the top section here. So exons, they will be expressed. I kind of think of I try to remember that as exons are extroverts. They are going to be expressed. They will code for a sequence of amino acids. Introns, like introvert, they're not going to be expressing themselves. They're in fact going to be taken out of the finished messenger RNA product. So right now we are still at the level of DNA. So let's go from DNA. Here's the next, just less labeled strand of DNA, same thing. And we have an exon, we have an exon, and an exon. So we'll say exon 1, exon 2, exon, so that'd be exon 3. Then we have these two introns here, intron 1 and intron 2. So essentially what's going to happen is these introns will eventually be cut out, and then exon 1, 2, and 3 will be joined to each other. So transcription occurs, and the whole length of the gene is transcribed from start, where the promoter is, to the end, where there is a termination sequence. Now we are still kind of color coding the introns as a darker color. So intron here, and then we have those three exons, exon 1, exon 2, and exon 3. And the introns are going to be cut out, and here at the very bottom, the finished messenger RNA product is going to consist of exon 1, exon 2, and exon 3, all joined to each other. So why on earth would a cell do this? One of the functions is that a single sequence of DNA can be processed in different ways. So it's transcribed into messenger RNA. So right here, that's the messenger RNA. And then depending on how the cell processes it, so maybe in one circumstance, the cell will take out all of the introns, and we will get one polypeptide protein product. In another circumstance, maybe in a different cell, the cell is going to take out half of the introns, and we'll get a somewhat different polypeptide product. So in this way, we can code for multiple polypeptide products with one sequence of DNA. Translation involves ribosomes and tRNA. So tRNA is down here, and there's going to be a couple parts that you need to know. And well, actually really just one, and that is this, the anticodon right here. That's what we're going to be focusing on, because that anticodon is going to use the base pairing rules that we talked about with RNA to bind to the messenger RNA. So now we have two kinds of RNA. We've got tRNA and we have messenger RNA. They will bind to each other like so, U to A, and A to U if we switched them around, C to G, G to C, etc. They will bind to and recognize each other in that manner. The ribosome will facilitate this process. Here we go with translation, and it takes place in 3 phases: initiation, elongation, and termination. Pardon me. Before we go over this, I would recommend taking a look at the animation that is posted on Blackboard in this section, and it just shows translation in process. And once you see all the moving parts, it'll be a lot easier to hear to understand what I'm talking about here. So you might want to pause this video and take a look at that animation. Translation. So here we go. This is all of it in summary. And what I want to go over now is just labeling the parts of this process so that when we talk about them, you know what we're talking about. Okay, so all of the purple fellas here, those would be amino acids. And amino acids are going to bind to a specific tRNA, or transfer RNA. Here is one such molecule. Here's one transfer RNA, here's another transfer RNA, and each amino acid has its own specific transfer RNA. Down here, we can see that we have a transfer RNA in blue, kind of a tube-like looking structure, even though they are in fact a sequence of single-stranded RNA, like so. This is more like what a tRNA looks like. The tRNA down here is going to bind with its anticodon to codons in messenger RNA, and that's going to happen in a complementary way. So the anticodon of this tRNA here is CCG, and its codon that it will bind to is GGC. So take a look here. They are complementary. So that is how the ribosome ensures, or how the cell ensures, that the correct tRNA comes with its correct amino acid, and this is the genetic code. So that when we look up in that big table that was shown earlier, titled the Universal Genetic Code, this is how we ensure that GGC, that codon there, that GGC, is going to always code for this amino acid, glycine. How? Because GGC is complementary to CCG in the anticodon of this tRNA that always carries glycine. Let me see. Is there anything else I wanted to mention? Right. This is the growing polypeptide. And essentially what's happened is each of these tRNAs will bring an amino acid, and the ribosome will then catalyze the formation of a peptide bond between neighboring amino acids. The transfer RNAs leave, and they don't have their amino acid anymore. They can be reused. Initiation is the first part of translation. So number one, we are going to have the small ribosomal subunit first bind to a tRNA. What tRNA is it? Well, it's going to be the first tRNA in the sequence. So whatever tRNA is complementary to the first codon in the messenger RNA. So there's a start codon here, which is AUG. That's the only one that I have memorized because it's always the first one, AUG. And note that it is complementary to the anticodon in this tRNA. This tRNA has its anticodon of UAC that is complementary to AUG. And you can pause that and just ensure that you understand the complementary base pairing rules. After this tRNA, which always carries methionine, has bound to the start codon and the small ribosomal subunit has facilitated all of this, then the large ribosomal subunit binds, and essentially the initiation complex, or everything we need to start this whole process, is now ready to go. Elongation is when the ribosome is going to move along the messenger RNA and build the polypeptide. Note that there's a catalytic site here, and also note that the animation and one of the first slides I showed you showed that there were 3 tRNA binding sites. You may simply disregard them. They're not pertinent. The differences are not pertinent for this course. So here we go. The 2 sites that are here. Basically, what I want you to know is this. So in blue, we have a tRNA here, and we also have a tRNA here. Each of the tRNAs has its own amino acid. The first one has MET, the second one has VAL, stands for valine. And how do we know that the correct amino acids are brought? We know because the tRNAs, T stands for transfer, they have an anticodon which is complementary to the messenger RNA. So messenger RNA is down here, tRNA is in blue, and we see that their anticodon, I'm going to circle this one here, CAA is complementary to GUU. And so that's how the genetic code ensures the correct amino acid is added. This catalytic site here, that is where we're going to have the formation of a peptide bond between neighboring amino acids. So MET and VAL, take a look over here. The ribosome catalyzes the formation of a peptide bond between MET and VAL. After that, we get elongation. So we're going to elongate this, the polypeptide. How do we do that? The ribosome moves over one space so that there's now an empty location for another tRNA with its amino acid to enter the ribosome. So the next tRNA happens to be carrying HIS. I think that stands for histidine. And again, same thing. The ribosome will catalyze the formation of a peptide bond. So now we have MET, VAL, HIS in that order. And this just keeps going until we get to what is called a stop codon. So look over here, stop codon. Now, what does that mean? Stop codon is just It's a codon that says stop, basically, and there's not a tRNA that binds to the stop codon. It's like sort of another thing that binds to the stop codon, and just it's kind of like putting a rock in the road that the train's going down, and the train's just got to stop and get off the tracks. So the ribosome here, big unit, small unit, they both break off and disengage with the messenger RNA, and now we have this completed polypeptide. It's a pretty short one, 6 amino acids long. What's the length of a typical protein? Hundreds or thousands of amino acids long. So this process is pretty long, and do mistakes happen? Yeah. When I took a more complicated course in university about this, my professor thought, and he admitted he didn't know either, that probably like 80% of these polypeptides that are made incorrectly and are basically just tossed in the quote-unquote trash of the cell. So we now should feel like we know a little bit more about where this protein comes from, the CFTR protein. It is coded for in DNA in groups of 3. That is transcribed into codons in messenger RNA. And then if there's no mutation, then we get a normal CFTR protein like so. It does what it's supposed to do. And if we get a mutated one, could mean that maybe some amino acids have been changed in some way. So we're going to look at mutations a little later on in this chapter and come back to looking at cystic fibrosis also. Transcription and translation in summary. I'm just going to let you pause the video and read through these things and make sure they all make sense to you. These are simply the main points of what goes on in transcription and translation. So you can pause the video and take a look. In diagram, this is what happens in transcription and translation. In transcription, the DNA double helix, as we can see, is unwound by RNA polymerase, and RNA polymerase synthesizes a pre-mRNA strand that is complementary to the template strand. This is the template strand here. And the pre-mRNA is processed, meaning the introns are taken out, the exons are linked together. The mRNA, now completed, leaves the nucleus and associates with a small ribosomal unit, the large ribosomal subunit, and then these tRNAs show up, each bringing with them a single amino acid that is specific to their codon or their anticodon as well, and a polypeptide is synthesized. Mutations and proteins. Recall that mutations are changes in the base sequence of DNA. Remember, the main types of point mutations in DNA are substitutions, deletions, and insertions. So we'll go over each of them and we'll look again at what the effects will be on the polypeptide now that we know how polypeptides are synthesized. Nucleotide substitution. So here, an incorrect nucleotide takes the place of a correct one. If the wrong base still codes for the same amino acid, then there will be no effect on the protein. If the wrong base codes for a different amino acid, then the protein could be mildly or even severely affected. It depends on what amino acid it is. Generally, it will be, I would say, no effect to very little effect. If the change in base codes for a stop codon, then the protein is not going to be complete and will almost certainly be non-functional. So remember, let's go through a couple. For example, let's look at phenylalanine here. If we had a mutation that caused the messenger RNA to go from UUU to UUC, it would have no effect on the polypeptide. We would still produce phenylalanine at that location. On the other hand, if we went from, say, let's look at tyrosine here, draw your attention there. If we went from UAC to UAA, so we substitute a C or an A for a C, then that would produce a stop codon, and a stop codon means that's the end of a translation, and it is going to result in a much shorter polypeptide, probably going to be a total loss of function for that protein. The other kind of mutation, let's look at histidine and glutamine just as examples. We could use a lot of examples. But if our original sequence was CAC and we had a substitution mutation for the second C, so now it's CAA, then that would result in glutamine showing up where histidine originally showed up. And probably it wouldn't be a very severe mutation, might not have an effect, probably wouldn't. Look at a couple more examples. So on the top, we have normal DNA, and it is going to produce a normal polypeptide. Looking at just the diagram here, notice that these black lines there are kind of separating our codons. So there's a separation between a codon, there's another separation, and there's another separation. So here is one codon, essentially. AAA codes for UUU, and then ATA codes for UAU, and CGT codes for GCA, etc. So they've organized this just to remind you that everything comes in groups of 3. A silent mutation. So that's the first. I already kind of explained what a silent mutation is. And that is when, so looking at the normal one, the first codon is AAA, and it's been mutated to AAG here. So AAG is going to code for UUC in the messenger RNA. So it's just a change of 1, but it turns out that UUC also codes for phenylalanine. So phenylalanine, PHE, it's still the same amino acid as in the normal polypeptide above. So phenylalanine, mutation occurred, phenylalanine. That is called a silent mutation. So silent mutation. We don't hear anything, and we don't see anything. A missense mutation is when we're going to go from one amino acid to a different amino acid. So let's look at the bottom here. Missense mutation. Take a look at the third amino acid, that is GLY, codes for glycine. And here's our codon that we're interested in here. So it's the third codon. In the DNA, it is it's been mutated to CCT. Let's look all the way at the top. I'll erase some of this stuff so it's a little more clear. So up at the top. CCT used to be CGT. So CGT codes for alanine, and CCT codes for glycine. So this is called a missense mutation, and it results in the substitution of one amino acid for another amino acid. Again, generally pretty minor. Last one, a nonsense mutation, and that is over on the right here, C, nonsense mutation. And this is when we get a stop codon produced. So the original codon was, what was the original? I'd have to look at the second one. So back up at the top left of the screen here. Yeah, so messy. So much easier when I can just point instead of having to draw. So the second codon here that used to code for tyrosine, TYR, it was ATA, and now if we look back over here, it is ATT. So that ATT codes for messenger RNA that is UAA, which is a stop codon. So that means no further codons will be read. The ribosome will disengage, and that'll be it. So phenylalanine is the only amino acid in this polypeptide. Going to be a total loss of function. The polypeptide is totally misproduced. Mutations and proteins. So now insertions and deletions, usually much more severe mutations. Nucleotides are added or subtracted from a gene. These types of mutations are really bad because the reading frame of the codon become changed. So they can also be called a frame shift mutation. Example, here I'm going to use yellow. So remember that codons and triplets in DNA come in 3s. So the dog saw the cat. So it's in a frame. So if we, for example, deleted what are we going to delete here? Let's just start reading through it. So the first 3 is the, second 3 is dog, and then AWT, what's up with that? Ought. So we deleted an S. An S was deleted. So this third codon makes no sense anymore. It's AWT. We completely misread it, ought, and then it's a heck, and then at, and then the last one is not even 3, so that wouldn't even code for any amino acid. So the idea here is you delete one base and everything shifts over after that base. So in the example above, notice how the deletion of one letter or base causes all the words or codons after it to be read incorrectly. The resultant protein has a very different amino acid sequence and is almost always non-functional. Sometimes this can also code for the production, or not code for, but result in the production of a stop codon somewhere along the line down from the deletion or insertion mutation. And of course, that'll also be really serious. Cancers can be a result from major mutations like these. Mutations in proteins. So again, at the top we have the normal strand of DNA. It's normal. That's the most common one. And we get a frameshift mutation. So in this case, we're going to have an insertion. So the first codon or base trip or DNA triplet, still the same, AAA, but then after that, a T was added. So the second codon, instead of becoming, let's see, what was it above here? Above it was ATA, but now it is TAT. Notice the ATA here has just been kind of shifted downward one by one nucleotide, and so that shifts all of the resulting codons after that. So you add something or you delete something, it's going to be a major mutation. Where was it? Where was I here? E, the bottom one down here, this is a deletion mutation, so we take out a T. So it used to be, the second codon used to be A, T, A, and now it's going to result in all of these moving down. So the first codon would be, or the second codon would be A, A, C. So this group of three. Then the next one would be G, T, G. So everything is changed in a deletion or insertion mutation downstream of where that nucleotide was added or taken out. Cystic fibrosis, many different mutations, and they all cause cystic fibrosis. Some mutations cause the protein to be broken down, and others simply non-functional, like in this diagram. It is still in the cell membrane, but it's not doing anything. And you might wonder, why is it that there are so many different mutations that all cause this same allele or same disease? And the reason for that is we have to look back a long time ago, back to this mutation typically happens in people with European descent. So people with East Asian descent, Sub-Saharan African descent, really going to be pretty rare for them to get this. And it turns out that although it's really not great to have this, because if you have two copies of it, then you have cystic fibrosis. But if you have just one copy of it, what it means is you're going to have some functional proteins and also some non-functional proteins. And you might think, well, that's not as good as having two functional proteins, but here's the thing. This protein, the functional one, also serves as a binding site for several really severe types of bacterial infections. So if you have less of them, then you're much more likely to not get sick from certain types of bacterial infections. I believe the two most common that this mutation kind of gives you a little protection from are cholera and also typhoid fever. This protein is not just in the lungs as well. It's also in the intestines and the pancreatic duct. It's kind of all over the place in the body. And it means that bacteria just can't bind to it if it's mutated. So there's kind of it's like well, you win some, you lose some. If you have this mutation, you're kind of protected from cholera, typhoid, but if you have two copies of it, then you have cystic fibrosis, which is really negative. So we ask ourselves, why is this so common? And it's pretty common in populations having European ancestry. It's common because if you are lucky and you just got one bad gene for cystic fibrosis, you don't actually have cystic fibrosis, but you have resistance to these terrible diseases. I'm not going to be quizzing you on the evolution of this stuff because it's not in the book, but you might be wondering, why do we have these mutations? And it turns out that sometimes a mutation can be really bad if you have two copies, but really pretty good if you just have one copy. How are human genes regulated? So the human genome contains 20 to 30,000 genes. Why is it that we don't know, have a more precise number for that? Depends on what you consider a gene. Is one gene just a segment of DNA? What if the messenger RNA is processed somewhat differently? What if the protein is modified in some way? Does that make it a different gene? And there's also a huge amount of DNA that doesn't code for a polypeptide, but still results in a change in phenotype or a change in what the person looks like. So that's why we're really not sure, and it depends who you ask. A given cell expresses, or rather transcribes, only a small number of genes. Some genes are expressed in all cells, and other genes are expressed only in certain types of cells at certain times in an organism's life under specific environmental conditions. A couple examples are milk protein that's only going to be produced in cells that are involved in producing breast milk, so obviously not going to be produced in your fingertips, for example. Bruises, when cells are damaged, the response of surrounding tissues is to turn on a whole bunch of genes, turn off other genes, and that is what causes them to a bruise to undergo all of these color changes, or at least that's one of the things that causes all these color changes. Gene regulation. So number one, transcription. Cells can control the frequency of transcription. They can never transcribe a gene. They can be always transcribing a gene, and it depends on what cell type it is. For example, really clear example, a cell that is involved in the perception of light in your eyeball is going to use totally different genes than a cell that is responsible for, say, producing sex cells like egg cells and sperm cells. You don't want your eyeballs producing egg cells and sperm cells. They use different genes, and that is the use of those genes can most effectively be controlled by saying, are we going to transcribe this gene or are we not going to transcribe this gene? Number two, mRNA processing. Different mRNAs may be produced from a single gene. That depends on how many exons are removed and a variety of other things. mRNA processing. We can process the message a little differently. Translation. Cells can control the stability and rate of translation. So this is not so much a turn the gene off or turn the gene on the thing. It's more of a increase how much the gene is used or maybe decrease how much the gene is used. This is more for fine control, such as there's a little bit too much of this enzyme, we need a little less. So we're going to translate this protein, translate this mRNA a little less. Modification. Cells can regulate a protein's activity by modifying it. This will happen in the rough ER or the Golgi body most commonly. Can also happen in the cytosol. Point being, we can modify a protein after it's been produced. Last one, degradation. So we can just break down proteins. So after they've been made, we can break them down, and that will essentially get rid of the effects of that gene, at least to some extent. Transcription factors. These are usually proteins. They bind to DNA and inhibit or promote binding of RNA polymerase. So this is a way to turn on or turn off a gene. Let's take a look at this diagram first. Remember that in DNA, there's a region called the promoter. And the promoter basically will promote the binding of RNA polymerase. When RNA polymerase has bound, it will then start transcribing the gene. Turns out there's a whole bunch of different kinds of, usually proteins, that can bind to the promoter and increase or decrease the frequency or that RNA polymerase binds to the promoter. So is RNA polymerase going to be binding to the promoter and transcribing the gene constantly over and over and over? Or does it do it for a little while and then stop? Well, all of these different transcription factors, things that bind to the promoter, can increase or decrease the frequency RNA polymerase will bind. So this is how a cell can really finely control how much a gene is used. Perhaps, for example, if all four, 1, 2, 3, 4, transcription factors are bound, and RNA polymerase is just binding and binding and binding and transcribing and transcribing and transcribing. So a lot of expression, lots of gene expression. Then maybe if we don't have the fourth transcription factor, perhaps RNA polymerase will bind still, but less frequently. And then if we delete transcription factor number 3, maybe RNA polymerase now will just bind even less frequently. So we can have very fine control of how often transcription occurs. Example up here is estrogen, which you've all probably heard of. It's the most important female hormone, coding for many traits that are specific to females, although it also is very important in males as well. Estrogen will basically, I'm just going to say, activate a transcription factor. We then get DNA binding and then transcription of albumin, for example, a protein that is related to the response of the cell to estrogen. So estrogen can therefore, it's a hormone and it can turn on genes. Epigenetics. Only thing I want you to know about epigenetics is this: gene regulation that can be inherited from one generation to the next. Most famous example would be the Dutch famine that occurred right after World War II. Huge number of people starved. It was after World War II. The world was in tatters and there was not enough food in Holland. And there are about 20,000 women who are pregnant, and they had all these changes go on in their bodies, and it turns out that the gene regulation coded for, well, various diseases. One of them was diabetes. They became later in life more at risk of diabetes, and then their daughters and sons were also at an increased risk of diabetes, and even their grandchildren were at an increased risk of diabetes. This is epigenetics. Epi means above, genetics. So multi-generational gene regulation. Lastly, really scary story to tell around the campfire. If you're a DNA molecule, and when he looked at his strand the next morning, one of the bases was gone. Ugh, terrible joke, but well. All right. Hope you enjoyed this PPT, and I'll see you next chapter.